Hello, everybody. So we're going to discuss Chapter 2, Molecular Interactions for Human Physiology. Okay. So we're going to talk about molecules and bonds and covalent, non-covalent bonds, interactions, and we're going to also look at some protein interactions as well, okay? And some specifics about how proteins bind and things like that. So when we look at the molecules and bonds, right, especially the molecules that's going to be inside our body, right, we're going to have organic molecules, right? Our organic molecules are going to contain carbon, right? So that carbon is going to be very important. That will indicate if something is organic or not, right? So the biomolecules that we have in our body, right, they're going to make up the majority of us, right, of our being. And those are going to be the carbohydrates, the lipids, the proteins, the nucleic acids. So remember these are going to be the four major biomolecules, or the four major macromolecules that's going to be in the body. Okay, and they're mostly going to contain carbon, hydrogen, and oxygen atoms. Okay, and their basic functions, we're going to use these for energy. We're going to use these as building blocks as well, right? As we break down, for example, carbohydrates and lipids, right, to get energy from them. And we use proteins, right? Nucleic acids, things like that, to build up, right, things, okay? And so lipids are going to be these biomolecules made mostly of carbon and hydrogen. They're going to have a backbone of glycerol, and they're going to have either 1 to 3 fatty acid tails, okay? And so these lipids, very important because they're going to be non-polar. All right, so they're not going to be soluble in water. They're not going to like to be in water. Okay, they're not going to solubilize. They're not going to want to interact with an aqueous solution. Okay, so our lipids, they're going to be divided into two broad categories. We're going to have fats, and these fats are going to be solid at room temperature, right? And so most of these fats are going to be derived from animal products or animal sources. And then we have oils. Right, oils are going to be liquid at room temperature, and these are going to be like plant-based lipids, okay, or plant-based oils, okay. Now, when we're looking at the structure of a fatty acid, so fatty acids are just these long chains of carbon atoms that are going to be bound to these hydrogens. There you can see how a long chain of carbon that's going to be bound to hydrogens, and it's also going to have this carboxyl group at the end, right, and that's going to be that acid group, okay. So we have saturated fatty acids and unsaturated fatty acids. So saturated fatty acids, they do not have a double bond, okay? Notice how there's no double bonds in this chain. But an unsaturated fatty acid, they have double bonds, right? So a monounsaturated fatty acid has one double bond, but a polyunsaturated fatty acid is going to have multiple double bonds, and it's going to be the double bonds between the carbons, okay? Notice how this carboxyl group, this is going to be a functional group, it has a double bond in there, but that's not associated with the actual fatty acid tail when we're talking about the double bonds within that chain, okay? So when we're forming a lipid, right, a lipid is made up of a fatty acid, which we can see right here, these fatty acid plus a glycerol backbone, okay? And this glycerol is just simply a 3-carbon molecule that makes up the backbone of these lipids, okay? So we have the glycerol, right, plus 1 fatty acid. This is going to be a monoglyceride, all right? Mono means 1. If you have a glycerol, right, glycerol backbone, plus 2 fatty acids, we're going to have a diglyceride. The most common one that we're going to see, right, more than 90% of the lipids are going to be in this form, and that's going to be a triglyceride, right? Triglycerides have that glycerol backbone and 3 fatty acid tails, okay? And so there are other lipid-related molecules, right? These are fat-soluble, lipid-soluble, right? They are non-soluble in water and aqueous solutions, right? So other lipid-related molecules, there's going to be these eicosanoids, right? These are going to be those modified 20-carbon fatty acids with a complete or partial carbon ring at one end and 2 long carbon chain tails at the other end, right? So an example would be prostaglandin or thromboxanes or things like that. So these are going to be regulators of physiological functions. We have steroids, right? Those steroids are going to be these lipid-related molecules whose structure include that 4 linked carbon rings. So look at these rings, right? These ring-like structures, right? That's going to be indicative of those steroids for sure. And we have cholesterol, right? That main steroid that we're going to have in the body, primary source of steroids. A lot of the steroids, like those sex steroid hormones, they're going to be derived from like this cholesterol backbone, right? And even using this cholesterol backbone, we can see how we can make cortisol, okay? And then other lipid-related molecules are going to be phospholipids, right? Phospholipids, they're going to have 2 fatty acid chains, right? They're going to have a phosphate group too, okay? And of course, they're going to have this glycerol. Right? So cholesterol and phospholipids, they're going to be important components of our cell membranes. So we talked about lipids, right? Carbohydrates, these are going to be the most abundant biomolecule. They're going to have their name from their structure because they're going to really be made up of carbon and water, right? Carbohydro, right? Carbon and water. So their general formula is going to be CH2O, and then you can have and on the outside, it's going to be the number of how much carbon, how much hydrogens, how much oxygens are going to be in this carbohydrate molecule, right? And that general formula just shows that for each carbon, there's going to be 2 hydrogens and 1 oxygen, okay? And so these carbohydrates, they can be divided into 3 categories. We can have monosaccharides, we can have disaccharides, or we can have polysaccharides, right? Monosaccharides, going to be those simple sugars, okay? The most common monosaccharides. These are going to be the building blocks, right, of these then complex carbohydrates. And they're going to have either 5 carbons, such as ribose, right? So ribose has 5 carbons in it, right? So 1, 2, 3, 4, 5, okay? And you can see deoxyribose, still a ribose sugar, right? A pentose type of sugar. 1, 2, 3, 4, 5 sugars, okay? Or you can have another type of monosaccharide, which is a hexose or a 6 carbon sugar, such as fructose, glucose, or galactose, right? So we have 1, 2, 3, 4, 5, 6, right? And you can kind of see in this glucose molecule right here how they have those carbons written. So wherever you see those lines, how I circled, those are going to be carbon atoms, okay? And even galactose is going to be another simple sugar, another monosaccharide, okay? Then you have disaccharides. So disaccharides are going to consist of a glucose plus another monosaccharide. Okay, so we have sucrose, which is like table sugar. So you have glucose bound, right, via glycosidic bond to fructose, right? Or maltose, you have glucose plus another glucose. Or you have lactose. Lactose is galactose bound to glucose, right? So these are disaccharides consisting of a glucose and another monosaccharide. So 2 sugar components, right? And then you have polysaccharides. So polysaccharides are going to be glucose polymers. All right, so many many sugar or monosaccharides connected together. Okay, and so all living cells will store glucose for energy in the form of a polysaccharide. Okay, whether it's going to be glycogen, right? So we animals, humans, right, we store glucose in the form of glycogen. Glycogen is a polysaccharide because it contains many glucose units, right? So poly means many, right? Poly, many. Saccharide is sugar. So many sugar units, okay? And so another example would be plants, right? Plants, they store glucose in their polysaccharide form in the form of cellulose and starch, okay? And then we have amino acids, right, and proteins, okay? So let me just go back for just a second because I want to point out that for lipids, right, one of the major biomolecules, right, lipids, carbohydrates, proteins, nucleic acids. For lipids are going to be the macromolecule, okay? But their building block is going to be the fatty acids. The fatty acids will make up, these are the building blocks of lipids. And for carbohydrates, we have these monosaccharides, these disaccharides, right? These are going to be the building blocks of these carbohydrates, right? Carbohydrate is a complex sugar. So you take up these, simple sugars, you add them together, and you make a complex sugar, okay? So those are the unit or the building blocks of carbohydrates, okay? And now when we talk about proteins are going to be the macromolecules or the biomolecule, right? But what makes up a protein? It's going to be the amino acids, right? The amino acids will make up proteins. So all amino acids, they're going to have a carboxyl group, right? So they're going to have a carboxyl group, and they're also going to have an amino group, right? They're also going to have a hydrogen attached to that central carbon. And what's going to be different is this R group. It's this R group that's going to really identify what kind of amino acid it's going to be. So this R group is going to be different, okay? The R groups, they're going to differ in size, shape, and their ability to form hydrogen bonds or ions, right? So certain amino acids like glycine, right? Glycine is going to have a different R group than alanine, which is going to have a different R group from tryptophan, right? So they're going to have different R groups, right? So this is going to be the identifying factor that will make it a specific different amino acid. But all amino acids are going to have this carboxyl group, they're going to have this amino group, and they're going to have this hydrogen, all bound to that central carbon, okay? And so to connect amino acids, right, how we are able to build and make a protein, right, we are able to connect these amino acids, right? So in order to connect them, we form this peptide bond. It's this peptide bond that links two amino acids together, okay? So remember that amino acids are going to be linked by peptide bonds, okay? And so, here are some examples of amino acids in natural proteins, right? We have arginine, aspartate, cysteine, glutamate, glutamine, glycine, tryptophan, tyrosine, right? And these are their 3-letter abbreviations with their 1-letter symbol, of course. But there are a few amino acids that do not occur in proteins but have important physiological functions. Those are going to be the homocysteine, right? That's going to be a sulfur-containing amino acid that's in excess, and that's going to be associated with heart disease. You have gamma-aminobutyric acid, or GABA. That's going to be a chemical made by nerve cells that's going to be in the brain. And you have creatine, right? And creatine is going to be a molecule that stores energy when it's bound to a phosphate group, okay? Like phosphocreatine. So amino acids link up via peptide bonds to make proteins, right? Proteins, when we're looking at a protein structure, right, we have 4 primary folds or 4 primary structures of proteins. We have the primary structure, okay? The primary structure is just simply going to be the sequence of amino acids, okay? It's going to be the sequence of amino acids, okay? And as you can see, the sequence, right? This is just a general sequence of amino acids, okay? So the sequence of amino acids in a peptide chain, they're going to be the primary structure, right? And then the secondary structure, this is where we have some hydrogen bonds being created, right? And this is going to be between adjacent, chains or loops, right? So this is where we have some bonding taking place, right? So this is where we're creating the alpha helices and the beta sheets, okay? And then that protein is going to fold on itself a little bit more, creating more bonds, right? In between these chains, okay? And so you have these alpha helices and these beta sheets, they're folding on each other, creating in this 3-dimensional shape, right? So this tertiary structure, we have this 3D shape now. And then their final folding, this is called the quaternary structure. This is where multiple subunits will combine to form this one whole complex globular-like protein, right? And so we give this example right here, hemoglobin, and we know hemoglobin, or well, adult hemoglobin, right, is made up of 2 alpha chains and 2 beta chains, right? And so, for example, all of these chains coming together, right, folding up together, will make this quaternary structure of the protein, okay? And so here, you can see, okay, well, it's just the 3D structure. This is just the alpha chain, the beta chains, right? And now they're actually coming together to form this quaternary structure of the protein structure, okay? And so our last macromolecule, right, or biomolecule, right? And remember that biomolecules, they play an important role in energy and information transfer, right? It's going to be nucleic acids, right? So this would be nucleic acids, right? And the building blocks of nucleic acids are these nucleotides, right? And these nucleic acids are just our DNA, right? And our RNA, our genetic material. But it's the nucleotides that make up these nucleic acids. These are the building blocks of nucleic acids, right? So a nucleotide, as we can see here, let's make it a little bit bigger. A nucleotide is going to consist of a phosphate group, one or more phosphate groups that we can see here. It's going to also consist of a 5-carbon sugar, such as that ribose, remember, or ribose or deoxyribose, either or. And then we also have this nitrogenous base. So these are the 3 components of a nucleotide: a phosphate group, a sugar group, and a nitrogenous base. All right. So we have 2 different types of nitrogenous bases, right? We have purines, which have a double ring structure, right? So it's 2 rings right here. These are purines. And we also have pyrimidines. Pyrimidines have a single ring structure, so it's just 1 ring. So there's 2 types of purines, right, within our DNA. There's adenine and guanine, right? Adenine and guanine. And then you have the pyrimidines. These are cytosine, thymine, and uracil. Okay? Those are going to be the nitrogenous bases that make up these nucleotides, right? So it's going to have either or. It's going to be an adenine, a guanine, cytosine, thymine, or a uracil, right? Added to one of these type of sugars. And it's either going to be a ribose or it's going to be a deoxyribose. And remember, D means without. Oxy means oxygen. So we're going to be without an oxygen. Specifically, we're going to be without an oxygen here at this carbon right here. Notice how this carbon has that oxygen right there. This carbon right here doesn't. That's what makes it deoxyribose, right? So again, these nucleotides are going to contain a nitrogenous base. They're going to contain a 5-carbon sugar, and they're also going to contain a phosphate group, right? They're also going to contain that phosphate group, right? It could be 1 to 3, basically. Okay. So, and remember I said 1 to 3, right? So a single nucleotide molecule, right, have basically 2 critical functions in the human body. Number 1, capture and transfer energy in high energy electrons or phosphate bonds. And number 2, aid in cell-to-cell communication, right? So that CAMP molecule, right, the CAMP, that's going to be important for cell-to-cell communication, how cells will communicate with each other. And then we have these energy capture and transfer molecules. This is how we are able to capture and transfer energy, right, within the cell, okay? And that's going to be in the form of FAD, NAD, right? And again, it's just adenine, ribose, phosphate groups, and we had riboflavin, or for the NAD, we're going to add nicotinamide, right? But the more specific ones that you'll really be seeing, though, is going to be like ADP and ATP. ADP is, again, we have adenine as the base, ribose as the sugar, and 2 phosphate groups. Adenine diphosphate is what it's called. And then ATP, adenine triphosphate, because we have 3 phosphate groups. Again, we still have adenine as the base, ribose as the sugar, and 3 phosphate groups attached to this. And this is going to be a high energy yielding molecule. We're able to throw off one of those phosphate groups, basically, right, as energy to support a reaction. Okay, it's those phosphate groups that we can absorb or give away that we're able to transfer energy or transfer electrons, okay? And so within these nucleic acids, right, remember nucleotides make up nucleic acids, right? So a nucleic acid is just a polymer of nucleotides, and they function in information storage and transmission, okay? These nucleotide chains will form the strands of DNA and RNA. And so when we're looking at a nucleic acid strand, right, we'll notice that it has the phosphate group, right? It has the sugar group, and it has the nitrogenous base. But I want to bring to your attention the base pairing, right? Within your DNA, these bases are paired, right? So bases on one strand form hydrogen bonds with bases on the adjoining strand, right? And these bonds will follow specific rules because purines are larger than pyrimidines. And remember, purines are double ring structures. Pyrimidines are single ring structures, right? Because of the space limitations, right? We're always going to pair a purine with a pyrimidine, right? So guanine forms 3 hydrogen bonds with cytosine, right? So 1, 2, 3. Guanine forms 3 hydrogen bonds with cytosine. And adenine forms 2 hydrogen bonds with thymine or uracil, right? So A to T, 2 hydrogen bonds, right? So let's write this down. A to T, 2 hydrogen bonds. A to U is 2 hydrogen bonds. And G to C, we're going to have 3 hydrogen bonds. Okay? And so in DNA, you'll realize that we have adenine, thymine, guanine, and cytosine. But in RNA, right, we have adenine, uracil, guanine, and cytosine. So the difference between, or one difference between DNA and RNA is we are replacing the thymine that's in DNA with a uracil, that's going to be in RNA. Okay, that's going to be very specific, okay? And this is a great key right here that you can focus on too, right? And I also want to point out that in DNA strands, in these nucleic acid strands, they're going to run anti-parallel, right? So the 3' end of one strand is going to be bound to the 5' end of the other strand, and you can see 5' end of this strand is going to be bound to the 3' end of the other strand. And that's just the anti-parallel nature of our DNA strands and how they're going to be replicated, okay? And so this is just another image that I wanted to show you, just base pairings, again, to show you how adenine binds with thymine via 2 hydrogen bonds, and guanine binds with cytosine via 3 hydrogen bonds. Okay, so remember that for sure. And these DNA strands are gonna run in anti-parallel nature. Okay, and again, the building blocks of DNA, it's going to be a nucleotide. These nucleotides are going to be made up of a sugar, phosphate, and a base, right? Makes up a nucleotide. Okay, and again, we can have adenine as a nucleotide, we have thymine as a nucleotide, we have guanine, cytosine. These are all nucleotides, so uracil. Okay, again, just a quick recap. The 4 major biomolecules, or 4 major macromolecules, are going to be lipids, carbohydrates, proteins, and nucleic acids. Okay, so biomolecules, they don't have to be purely one kind of molecule though. So we have conjugated proteins, right? These are proteins that are going to be combined with another biomolecule, such as lipoproteins, and these are going to be proteins that transport lipids, right? We have glycosylated molecules, and these are going to be molecules that are attached to carbohydrates, right? And so we have some examples of glycoproteins or glycolipids, right? These glycoproteins and these glycolipids, they're going to be very important because they're going to be components and structures of the cell membrane, and they're going to be important for cell-to-cell communication and cell signaling and things like that, right? But biomolecules, they're going to be made up of repeating units of polymers, okay? But again, we can have lipoproteins, proteins that transport lipids, or proteins that are going to be associated with lipids, right? like proteolipids or lipoproteins rather, right? They're going to be associated with like lipid component and a protein component. Just like how we have these glycoproteins and glycolipids, like how a sugar is attached to a protein or sugar is attached to a lipid, right? We have these conjugated molecules, okay? So I want to bring your attention to functional groups. There are functional groups, right? And these functional groups are going to be combinations of elements that occur frequently in biological molecules. They're going to move between molecules as a single unit, right? So these amino groups, right, it stays together, right? So you can aminate something, like add an amine group to something, like another molecule, or you can deaminate something, like so in the form of deamination, right? That's a process where you're taking away an amine group, right? And if you're taking away this amino group, you're taking this whole group together, right? Because this whole group, it comes as one. And the same thing as a carboxyl group, right? It comes as one. Okay? They're either added to another molecule or they're taken away from a molecule all together. It's a functional group. Or we can add a hydroxyl group to a molecule, or we can take a hydroxyl group away from a molecule. Same thing with a phosphate group, right? So you can phosphorylate something, right, in the form of phosphorylation. That's the addition of a phosphate group. Or you can dephosphorylate something in the form of dephosphorylation, right? That's the removal of a phosphate group. But remember, these functional groups, they move between these molecules as a single unit, okay? So electrons, they're going to have four important biological roles. They help to form covalent bonds, and this is the sharing of electrons between atoms, right, to form molecules. Electrons will also act as ions, right? And ions are going to be atoms or molecules which are going to gain or lose one or more electrons, right? So they're going to become electrically charged, right? And this is going to be the basis for electrical signaling in the body, right? So you lose an electron, you're going to be more positively charged. You gain an electron, you're going to be more negatively charged. So things like that, right? And we have high energy electrons, and these are going to be electrons in certain atoms that can capture energy from the environment or transfer those electrons to another atom, right? And we have, unfortunately though, free radicals. Now, these free radicals, these are going to be unstable molecules with unpaired electrons. And we don't want free radicals because they can cause a lot of disruption in our body, okay? So when we're looking at atoms and molecules, just a quick overview, right? We know that protons, right, positively charged, neutrons, neutrally charged, electrons, negatively charged. Protons are going to determine the element, right, or the atomic number. The neutrons are going to determine the isotope, right? The electrons, they're going to form the covalent bonds, create the ions, capture and store the energy, create those free radicals, right? We're going to typically find protons, neutrons in the center of the atom. We're going to find electrons on the outskirts of the atom, right? And the electron cloud, right? And so atoms, right, are made up of these protons, electrons and neutrons, right? And even though the proton is the atomic number, right, protons plus neutrons, they're going to be in the nucleus in the center of this atom, right? They're going to account for the atomic mass, so the weight of this atom, okay? And so we can see here, if we have two or more atoms that share electrons, they will form a molecule. So atoms make up molecules, and we can see this as a water molecule right here. We have one oxygen and we have two hydrogen atoms forming a molecule as they are sharing these electrons, okay? So when we're looking at covalent bonds between atoms, they're going to create molecules. So these covalent bonds, these are going to be strong bonds though, right? And they're going to require energy to make or break them. But these covalent bonds, so covalent bonds, the definition of covalent bonds is the sharing of electrons, okay? So we can have single, double, or triple bonds, and these could be either polar or non-polar molecules. So again, these covalent bonds, they're going to result when atoms share electrons. Remember that, okay? And these bonds will require the most energy to make or break them because they're going to require a lot of energy. It's hard to make or break these covalent bonds, okay? So we have non-polar molecules. Non-polar molecules have an even distribution of electrons, Okay, for example, we have molecules that are going to be composed of mostly carbon and hydrogen. They're typically going to be nonpolar, like this fatty acid, typically going to be nonpolar, right? And then we have polar molecules. Now, polar molecules, they have regions of partial positive or partial negative charge, right? So they have, this is the unequal distribution of electrons, okay? And you can see right here, let's bring this up a little bit. I'm going to highlight this right here, right? And so you have a partial negative charge from that oxygen, and you have a partial positive charge down here because of that hydrogen, right? So notice how we have all these electrons up here associated up here, but we have, you know, some positiveness coming from that hydrogen down in this region, okay? So you have an unequal distribution of this electrons, or in that electrical charge, okay? And that's what makes it polar. So nonpolar, you have an even distribution of electrons. Nonpolar, you have an uneven distribution of electrons in this molecule. And as a result, you have a partial positive charge and a partial negative charge, okay? So ions are going to be charged atoms. So we have cations, which they have lost electrons, so they're going to be positively charged. And you have anions. Anions, they have gained electrons, and these are going to be negatively charged species, right? So some examples of some cations would be sodium, potassium, calcium, hydrogen, and magnesium. Cations, right? And some anions would be chlorine or chloride, right? Bicarbonate, phosphate, and then sulfate groups. Those are going to be negatively charged, right? The gain of an electron, that's why you can see this negative charge associated with them, okay? So non-covalent bonds, right, they can facilitate reversible interactions, okay? And so ionic bonds, these are when ions form when one atom has such a strong attraction for electrons that it pulls one or more electrons completely away from another atom, right? And this creates that electron attraction between these ions, right? And these ionic bonds, atoms will either gain or lose electrons, right? And we also got to remember that opposite charges will attract. And hydrogen bonds, right? Hydrogen bonds are going to be these weak bonds. Remember I mentioned these are going to be, for example, these are going to be the bonds that hold the two DNA strands together, right? Like how guanine of one strand is going to be held with cytosine of the other strand via like 3 hydrogen bonds, right? So these hydrogen bonds, they're going to be weak, and they're going to require little energy to make or break them, okay? And hydrogen bonds will result from the attraction between a hydrogen atom and a nearby oxygen, nitrogen, or fluorine atom, right? And this gives the water surface tension, right? That's going to result from hydrogen bonding. And then we also have the other type of non-covalent bonds, right? So we talked about ionic bonds, we talked about hydrogen bonds, and we have van der Waals forces. These van der Waals forces, these are going to be weak and non-specific attractions between atoms, right? And so when atoms come in close contact, they exhibit these van der Waals forces, these van der Waals attractive forces with each other, okay? And so this again just goes over the non-covalent bonds, right? So ionic bond, right? These are electrostatic attractions between ions, right? Where we are gaining or losing an electron, okay? So you can see that, look at this sodium right here. It lost an electron to chlorine. So chlorine gained an electron. So because sodium lost an electron, it became more positive, and chlorine, it gained an electron, it became more negative, right? And again, hydrogen bonds are going to form between hydrogen atoms in a nearby oxygen, nitrogen, or fluorine, okay? And then van der Waals forces, again, they're going to be weak, non-specific attraction between atoms. Now, hydrophilic interactions, right? So we, hydrophilic is water loving, right? Hydrophobic is water fearing, right? Hydrophilic interactions, these are going to create biological solutions, right? So all molecules and cell components are either going to be dissolved or suspended in these solutions, in these biological solutions, right? So a solution is going to contain solutes that are going to be dissolved in a solvent, right? So we have water, right? Water is going to be an aqueous solution. All our biological solutions are going to be water-based, so they're going to be aqueous, right? So we have a solution, again, solutes dissolved in solvents. So the solute would be like an ion, right? That's going to be dissolved in the solvent, which is going to be the fluid, like water, okay? And so solubility will refer to the ability of a solute to dissolve in a solvent, right? So hydrophilic substances, these are going to be soluble in water, right? Such as polar molecules having an unequal distribution of electrons, right? Polar molecules, ionic molecules, these are going to be hydrophilic. They're going to be soluble in water. And we're also going to have hydrophobic substances, right? These are not going to be soluble in water, right? These are going to be non-polar molecules such as lipids, right? Equal distribution of electrons, right? Non-polar. They're not going to want to be in water. Okay. So it's just some solutions terminology. Solute, any substance that dissolves in a liquid. Solvent is going to be the liquid for which that solute dissolves in. And a solution is going to be a combination of that solute dissolved in that solvent. And however, the concentration of a solution is going to be the amount of solute per unit volume of that solution. Okay. So for example, when we're dealing with solutions, right, the mass or the weight of a solute before it's dissolved is usually going to be given in grams or milligrams, right? But the molecular weight is going to be calculated from the chemical formula of a molecule. Let me just do this so you know. The molecular mass is calculated from the chemical formula of a molecule, right? So this is the mass of one molecule expressed in atomic mass units, or more often in Daltons, though. So 1 atomic mass unit equals 1 Dalton. And so the molecular mass, right, again, calculated from the chemical formula of a molecule, is going to be equal to the sum of the atomic mass of each element times the number of atoms of each element, right? So for example, if we're looking at the molecular mass of glucose, and we have glucose formula is C6H12O6, right? Element, carbon. How many atoms does carbon have, right? Remember, it's going to be 6, right? And so what is the atomic mass of carbon, right? And so the atomic mass is going to be 12 atomic units, okay? And so we take the so we take the atomic mass is going to be multiplied times how many of them are there, like how many atoms are there, right? And so there's going to be 6 in this case of carbon, and so we take 12, the mass of all these carbons, of 12 carbons, the mass of 6 carbons, right? And we get 72. And so for hydrogen, right, there's going to be, how many hydrogens are there? There's 12 of them, right? So there's 12 of them, right? And what's the atomic mass of hydrogen, right? It's going to be 1, right? And so if we remember looking at the periodic table, hydrogen is number 1, okay? And so how many hydrogens are there? There's 12. So multiplying the mass times 12 of them, we're going to get 12. And so oxygen, right? How many oxygens are in there? It's going to be 6. So there's going to be 6 oxygens. How much does oxygen weigh? It weighs 16 atomic mass units. So we take 6 times how many of them? 16 atomic mass units, and we get 96. And we get the molecular mass of this molecule being 180 atomic mass units, right? Or the molecular mass of glucose would be 180 atomic mass units, okay? And so this just gives you the expression of volumes. Volume is typically usually going to be expressed in liters or milliliters, right? So typically we use liters and milliliters, right? So milliliter is going to be 1 to the negative 3. And then micro, we also talk about microliters too. Not necessarily so much in I've never seen, but in science for sure. But micro is going to be 1 to the negative 6, nano to the negative 9, pico to the negative 12. Okay. And so when we want to express a concentration, like the percent solutions, so in a laboratory or pharmacy, scientists, we can't measure out solutes by mole. Instead, we'll use the more conventional measurement of weight. And so the solute concentration may then be expressed as a percentage of the total solution or a percent solution. So a 10% solution means you have 10 parts of a solute per 100 parts of the total solution. So for example, like how 10% of 100 is 10, right? Or 50% of 100 is 50, right? So it's the same idea, right? But the weight volume solutions, these are going to be used for solutes that are solids. And these are going to be usually expressed as grams per 100 milliliter solutions, or milligram per deciliter, as we can see right here. So we have an example right here of solutions used for intravenous infusions, and these are going to be often expressed as percent solutions. So our question is, well, how would you make a 500 milliliter solution of 5% dextrose, right? So how can we make this? So we take 5% solution, right? So we know a 5% solution is going to be 5 grams of glucose dissolved in water to make a final volume of 100 mils, okay? That's basically 5% of 100, right? So you take 5 grams of glucose and you dissolve it in water up to making it a total of 100 mils. So it's that 5 grams that's dissolved in a total of 100 mils, okay? And so for 5 grams of glucose, right, we want to make a 5% glucose solution in 500 mils. So in order to do that, we take 5 grams of glucose. We know that 5% is 5 grams and 100 mils, right? And we're trying to find, well, how much of this glucose should we put in to make 500 mils, right? So we just do some mathematics right here, some cross multiplication, right? So right here, we have 25 grams per milliliter. Sorry, let me erase that. Let me actually do my math this time. 5 times 500, right? 2,500 grams per milliliter, right? Will equal, right, 100 mils times the grams, right? And we're trying to get this, right? So we divide that by 100 mils. What you do to one side, you got to do to the other side by 100 mils. These mils will cancel out. 100 goes into there, right? 25 times. That grams stays over, right? So we would have to put in, as we can see, 25 grams of glucose with water and add it to give it a final volume of 500 mils, okay? And we can do some more examples like that as well. So, right, let's look at these molecular interactions. So hydrophilic interactions, again, these are going to be water-loving molecules, right? So you can have molecules that have polar regions or ionic bonds. These are readily going to interact with polar regions of water, okay? This is going to enable them to dissolve easily in water. And so molecules that dissolve easily and readily in water are said to be hydrophilic. Molecules on the opposite end, right, that are nonpolar, right, these will not dissolve readily in water. These are said to be hydrophobic. Okay, hydrophilic, dissolving in water easily, readily. Hydrophobic, not dissolving in water easily and readily. Okay, so the molecular bonds, right? It's these bonds that are very important. So these bonds and these interactions, these are going to be critically important in determining the actual molecular shape of this molecule. Okay. So the covalent bond angles, the ionic bonds, the hydrogen bonds, van der Waals forces, all of these, right, they're going to interact to create a distinctive shape of a complex biomolecule, right? And we know that shape equals function, right? The shape will play a critical role in a molecule's function. Remember. Shape equals function. Can't stress that enough, okay? And so hydrogen bonds or hydrogen ions in solution, they can alter molecular shape though, right? So we have those free hydrogen ions, right? They can participate in hydrogen bonding and van der Waals forces, and that can change a molecule's shape or conformation, right? And so if we look at the last slide, it should have like, right? So looking at this legend right here, we have the disulfide bonds between these sulfur bonds, right? And we have these ionic repulsion, right? So look at these repulsion, right? When we have those lines right there, that's like repulsion, okay? And these hydrogen bonds, these are gonna be like the dots, right? So those hydrogen bonds, they will interact and they can help to form that molecular shape. But again, those hydrogens, those free hydrogens, they can participate in the hydrogen bonds and they can alter the shape of those molecules, right? The pH, right? pH is going to be very important, as we all know. pH is going to be the measure of the concentration of free hydrogen ion concentrations, right? So we know that a pH that's going to be less than 7 is going to be acidic, 7 is neutral, right? And more than 7, greater than 7 is going to be alkaline, right? So acids are going to be molecules that release hydrogen when they're dissolved in water. And we have buffers. Buffers are going to be really important in our body because buffers will moderate the changes in pH, right? So our bicarbonate anion, right? It's going to be an important buffer in our body that will moderate changes in pH, okay? So an acid, again, will contribute hydrogen ions to a solution, right? So for example, a carboxyl group, right, has that acid, that carboxylic acid, really, we call it too, right? Because it's going to contribute its hydrogen when it's in solution. So it's going to lose that hydrogen, all right? A base is going to be a molecule that decreases hydrogen ion concentrations in solution by combining with that free hydrogen, right? So for example, molecules that produce hydroxide ions, or this right here, in solution, these are going to be bases because that hydroxide ion can combine with that free hydrogen ion to form water, right, which we want in certain cases, right? Another ion that another molecule, right, that acts as a base is going to be ammonia. So NH3. NH3 will react with a free hydrogen ion to form ammonium, right? And so again, pH, the concentration of hydrogen ions in body fluids, is going to be measured by pH. pH stands for the power of hydrogen, right? And so this equation is going to be read as pH is equal to the negative log of a hydrogen ion concentration. And so this square bracket, remember that square brackets are just a shorthand notation for concentration, okay? So we just use the brackets by as a concentration of something, okay? And so looking at this pH scale, we know that normal pH of blood in humans is going to be like 7.4, 7.35, 7.45, something like that, right? Homeostatic regulation is going to be critical, though, because that blood pH less than 7 or greater than 7.7, completely incompatible with life. You're dead, right? That blood pH goes down or up, right? Hydrogen ions, too much hydrogen ions, too less hydrogen ions in your blood, you can die, okay? So that's why it has to be in that narrow range, okay? And we know that pure water has a pH value of 7. As we go down, right, down the pH scale, which means more hydrogen ions, right? Urine, tomatoes, grapes, vinegar, lemon, juice, stomach acid, these are going to be acidic species where we have pancreatic solutions, a little bit more basic, right? Baking soda, a little bit more basic, right? Soap, ammonia, chemical hair removers, of course, sodium hydroxide. These are going to be basic or alkaline solutions that have hydrogen ion concentrations lower than that of pure water, okay? So let's look at protein interactions. So proteins, they will act as so many things, right? Proteins act as enzymes, and enzymes are these biological catalysts that speed up chemical reactions. Proteins act as membrane transporters, right? And these are proteins in cell membranes that help move substances back and forth between the intracellular and extracellular compartments. Proteins act as signaling molecules, right? Act as a hormone or other signaling molecules. They act as receptors, where they bind signal molecules and initiate a cellular response. They bind proteins, right? These are going to be found mostly in the extracellular fluid. These are those binding proteins, right? They help to bind and transport molecules throughout the body, like ferritin, that's helping transport iron, hemoglobin, helping to transport oxygen and carbon dioxide. Proteins also act as immunoglobulins, right? And immunoglobulins are going to be these extracellular immune proteins, right, that help to protect the body from foreign invaders. And we have regulatory proteins, right? They turn cell processes on and off or up or down. Okay? And so proteins, right, they interact really via binding sites, right? And it's the binding sites where the binding actually takes place, right? So a ligand is any, molecule or ion that binds to another molecule. That's a ligand. But ligands that bind to enzymes, right, a molecule that will bind to an enzyme, and membrane transporters, these are going to be called substrates, right? So substrate binding to a enzyme, okay? Proteins are specific, right? And they're very selective. They are highly selective about the molecules that they bind, right? So proteins exhibit specificity, right? So the ability to specificity is going to be the ability of a protein to bind a certain ligand or a group of closely related ligands, right? So ligand binding require molecular complementarity. So a ligand and protein binding site must be complementary or compatible, right? So when we're looking at the binding of proteins to their ligand or enzyme to their ligand, right, we look at the model of induced fit. So this induced fit model of protein-ligand interaction, right? So this is where the protein's binding site and the shape of the ligand, they don't need to be, fit one another exactly, like an exact match. However, when the binding site and that ligand come into close contact, they begin to interact through hydrogen, ionic bonds, and van der Waals forces. And so that protein binding site then changes shape or conformation, right? It changes shape, conformation, right? To fit more closely to that ligand. So now they have a tighter fit. Kind of in a sense when you put your hand in a glove, right? And then that last little tug, you got to pull that glove down a little bit more. That's that induced fit, basically. And so when we're looking at this induced fit model of protein-ligand binding, in this model of protein binding, we can see that the binding site, the shape is not an exact shape that's going to match that L shape ligand, but once they come into close contact, once they come into close contact, this protein will undergo a conformational change, and then they'll bind. That binding will be ever more so better. Okay, so protein binding reactions, these are going to be reversible, right? So it's the affinity, right? The affinity is going to be the degree to which a protein is attached to a ligand, right? How much does this protein want to bind to that ligand? Its affinity to binding, right? And so the equilibrium is going to be the rate at which binding is going to be exactly equal to the rate of unbinding, right? So we keep it in an equilibrium. So we have these ligands binding to a protein, and we have ligands unbinding to this protein in a solution. That's going to be equilibrium, okay? And so we also want to mention that binding reactions, they were going to obey the law of mass action, okay? And so the law of mass action says that when protein binding is at equilibrium, right? So the rate of binding is equal to the rate of unbinding, right? The ratio of the bound and unbound components remain constant, okay? So we don't have more bounding or more unbounding. They are at equilibrium. Now the dissociation constant, right, indicate low binding affinity, right? So this ligand can easily just unbound to this protein, right? And that's because we have, number one, as we understand that this dissociation constant indicates affinity, we have competitors, and these competitors are related ligands that will compete for the binding site, right? And we have agonists, and these are competing ligands that mimic each other's action, right? So there are multiple factors that will affect protein binding, such as isoforms, So we have isoforms, and these are closely related proteins, and they're going to function in their function is going to be similar, but their affinity for ligands are going to be different, right? Such as like hemoglobin, like adult hemoglobin versus fetal hemoglobin, right? And there's other factors that affect protein binding, such as activation, right? This protein needs to be activated via proteolytic activation, cofactors. So some proteins, they need cofactors, such as like ions or small organic functional groups for them to actually be functional, for that protein to be functional. And we have modulation or modulators, right? These are those chemical modulators that will bind reversibly or irreversibly to proteins and alter their binding affinity, right? Or their binding activity. We have competitive, non-competitive inhibitors, right? Allosteric modulators, covalent modulators, right? So cofactors, right? These are going to be important because they're going to be required for ligand binding at a binding site. So some proteins, they need a cofactor. They need like a vitamin. They need like an ion there to actually successfully bind this ligand to it. Proteolytic activation. This is where we are able to convert an inactive to an active form, right? So this protein was inactive, right? But we cleaved it somewhere and we now have made it active, right? So such as, for example, like pepsinogen, the inactive form, we cleave it and we make pepsin, the active form, right? Angiotensinogen, right? We make angiotensin 1, right? It's inactive form, proteolytic activation of it, right? So cleaving some portion of it, and then we're able to put it in its active state, right? And then we have modulators, right, that will alter binding activity as well, right? So this is that proteolytic activation right here, where we just like cleave it right there, right? And now we have this active protein, right? We have cofactors. Cofactors are going to be important because this protein is inactive. That cofactor will bind to it, and now we can have the substrate binding to it. Okay, we have allosteric activators. So allosteric activators are these, it's going to be a modulator that binds to a protein away from the binding site. So these allosteric activators, they bind elsewhere other than the binding site. Okay, and so what happens is they bind to that protein, right? And they change the conformation shape of the protein, thus changing that binding site shape. And now that ligand can't bind in there anymore sometimes, right? So competitive inhibitors will block that ligand from binding at that binding site. So it competes for that binding site, okay? And we have allosteric inhibitors, right? Again, they're going to bind somewhere else, and they're going to inactivate that protein. That's, again, changing that conformation, right? Changing the shape of it. So now that ligand cannot bind. And we have multiple factors, right, that will affect protein bindings, right? Physical modulators. So remember this. Temperature will affect how a protein will bind and interact. pH, right? The body will regulate the amount of protein in our cells, where we will upregulate or downregulate it, right? The reaction rates also are a huge factor, as those reaction rates can reach maximum, right? And that's going to be with like saturation. So the concentration of the ligand. So how much ligand is there to bind, right? And are all of the protein binding sites, are they saturated? Are they all used up, right? So these are factors that will affect a protein binding, okay? Temperature, right? This active protein, right, will only be active, only be useful, right, in a certain temperature range, right? But you put that protein out of that temperature range, right, it starts to denature. And it's denaturing, it's not functional anymore. We can't use it, right? And so this is another factor, right, that I mentioned that's going to influence protein activity. The amount of protein and the amount of the ligand, okay? So this is just a overview of chapter 2, okay?